Hello, welcome to the Autism Analysis channel where we discuss autism research in an objective manner. In this video, we're going to talk about inheritance, various ways genes can pass from parent to child. First, some disclaimers. This is not a substitute for a genetics lesson. This only addresses models relevant to autism. Autism has multiple causes and only some are known. This talk includes theoretical models that may turn out to be false. De novo mutations. De novo means of new in Latin. They are not inherited from parents, but rather occur in an egg or sperm cell before or during fertilization, so the mutation is not present in either parent. If it occurs in a known autism gene, it can be identified in a genetic test. Once confirmed as a de novo mutation, the family can be confident it won't happen again in other children, since the odds of this are less than 1%. De novo mutations may be identifiable in less than 10% of cases, especially if no family history of autism or mental illness is present. They can be identified on genome sequences or microarrays. In rare situations, a parent may have a de novo mutation, but only in their germline cells, i.e. the area of their genitals. Because it is not present in the brain of the parents, they will appear to be completely unaffected. But because it is present in multiple germline cells, they may have multiple autistic children, and their children will appear to have the same de novo mutation if the mutation can be located on a test. A single gene has about a 50% chance of being passed on from any parent. If it is a recessive gene, it is unlikely to have an effect unless both parents carry the same recessive gene. Most of the genes that have been associated with autism so far do not seem to have recessive characteristics, however. So this model is unlikely to be applicable. Some genes associated with autism do not cause autism in every case. This has led to the idea that it is a combination of genetic effects. Some cases could be caused by inheriting genes from both parents who themselves are either mildly affected or completely unaffected by autism. We know the odds of a couple with one autistic child having another autistic child is about 30%, suggesting that a large proportion of cases of autism may be caused by a polygenetic effect. Fragile X syndrome is a specific form of severe autism that is caused by a mutation in the FMR1 gene, which lives on the X chromosome. This accounts for about 1% of autism, but this was the one that was most easily identified from genetic analysis. You can see a fragile-looking X chromosome if you were to observe it under a microscope. Because it is a sex chromosome, it is very easy to reason about the inheritance pattern of this sort of disorder. Since women have two X chromosomes, they are usually unaffected by this, but have a 50% chance of giving their sons the disorder, and every daughter has a 50% chance of being a carrier. A male with Fragile X is usually too disabled to reproduce, but he cannot give the disorder to a son, but would produce a daughter carrying the chromosome. Modern technology can easily detect the Fragile X chromosome with medical tests. Other genes are also associated with autism and appear on the X chromosome and can be reasoned about in the same way, but these would require genome sequencing to detect. Some have suggested that a number of autism cases could be related to mitochondrial DNA. Mitochondrial DNA is always inherited from the mother and does not change except through mutation. Any mutation in mitochondrial DNA would be carried by all children from the mother, but only her daughters would pass it on to the next generation. So to conclude, these are the basis of how genetic inheritance can work in autism, and we will cover other models in the future. Thanks for listening to this video.